Hello and welcome everyone. In this podcast we're going to begin our discussions on this term linkage. Up to this point, when we've been looking at Mendel's rules, we've been making assumptions that the genes that we're looking at are on separate chromosomes. So for instance, when he did a dihybrid cross and you had this homologous chromosome and say this gene on here was big Y, maybe this one was the allele for little y. And when he did a dihybrid cross with, say, round and wrinkled alleles, these happen to be on separate chromosomes. Now, the rules of assortment and segregation apply to chromosomes. So these two chromosomes segregate to separate gametes. They can either assort independently with this chromosome or with this chromosome. They don't necessarily specifically apply to the genes. It just so happens that the genes are on the chromosomes. But really these rules are all about the chromosomes. And so as long as the two genes you're looking at happen to be on different chromosomes, there's no problems at all. You would expect independent assortment of these two chromosomes and consequently the two genes. Well, then along came a few scientists. Morgan, remember he was the one that first won a Nobel Prize for his work in genetics. And then he had two students that worked with him, Sturtevant and Bridges. These were started off as undergraduates in his lab, and at least one of them was pretty much a C student, but went on to win many prizes for his ultimate genetic and molecular biology research. Sturtevant, while he was in Morgan's lab, he proposed that while he was in his lab, he observed that sometimes two genes did not assort independently. He reasoned that you could have a chromosome, and again this seems obvious to us, but at the time it wasn't that obvious. He reasoned that you could have a homologous chromosome where the two genes you're studying happen to be on the same chromosome. So maybe you're studying gene X and gene Y. Because they're on the same chromosome, they're not going to assort. The genes won't assort independently. The chromosomes will, but these two specific genes, they'll assort with each other. That's what he proposed. He also, and this is what we'll talk about a little bit later, Sturtevant proposed that these genes that are on the same chromosome, he came up with a way that we could map distances between these two genes. And we'll talk about that later on. So this really began the next several decades of research where they started to map genes to specific chromosomes. And they started to measure distances between genes. And ultimately that led to where we're at today with sequencing genomes, with manipulating genomes. So really a lot of this early work in molecular genetics began right here in Thomas Hunt Morgan's lab. I'm going to erase this right now so we can talk a little bit about some terms that we're going to need to know as we move forward. The first term is a term called synteny. This is when two or more genes are on the same chromosome. So if genes X and Y are on the same chromosome, we say that these two genes are in synteny with each other. We use this term linkage, and sometimes people will add a word to this and call it genetic linkage, but it's more common just to use the word linkage. Linkage means that we have two genes close enough to each other that they assort during meiosis. They assort with each other. So if genes A and genes B are on separate chromosomes, they're going to assort independent of each other. But if genes A and B are on the same chromosome, close enough to each other, they're always going to assort with each other. They are not going to independently assort. If gene A goes into a gamete, one gamete, gene B will always go into the same gamete because they're connected with each other, or they're linked with each other. Linkage group. Sometimes we call chromosomes a linkage group because a chromosome has many linked genes. And since they're on the chromosome, sometimes we just refer to that whole chromosome as a linkage group. Unlinked genes. And linked genes. Unlinked genes are on separate chromosomes. Linked genes are on the same chromosomes. I drew that on the last slide briefly, but let's go ahead and draw it again real quick. So if they're unlinked, you will have, well, this would be an example of unlinked genes. So we have two homologous chromosomes, and we have genes, say, A here, and genes B here. Two different alleles of the gene B and two different alleles of gene A, but they're on separate chromosomes. As a result, they're going to independently assort. We say they are unlinked. Now you could have linked genes, and again, all linked genes means is you have the same chromosome, but now the genes are on the same chromosome. So you may have genes C here and D. Now genes C and D are always going to assort with each other. Their assortment is dependent upon each other. Okay, so let's talk about crossing over. And to talk about this, let's talk about a homologous chromosome like so. And we're going to draw two possibilities with crossing over in these two chromosomes. And let's give some genes here. Let's say we have A here, and over here it's little a. Remember, these are sister chromatids, so they should be the same on both sides. And for this example, I want to make these chromosomes a little longer. Alright, I want to make a few points as we go forward. The first is that crossing over always occurs on a chromosome. The second point related to this is that crossing over doesn't always occur between the genes being studied. So over here, let's say we have crossing over here. Crossing over always occurs on the chromosome somewhere, but we're not going to see it because we're only going to be looking for crossing over events here, in between genes A and B. If it occurs anywhere else, we're not going to be able to see it. We're only going to see crossing over when it occurs here, in between the genes we're looking at. Now what I want to do is erase what I have down here and draw what the gametes are going to look like from each of these two situations. So first, let's draw the 4 gametes from this one on the right. And what you're going to get is this chromosome in this gamete with A and B on it. You're going to get this chromosome here with it. You're going to see these are all going to be quite similar. Now it's true crossing over occurred here, but we're only looking at genes A and B, and there's no change between those. And then this gamete and this gamete are going to have genes little a and little b. Since each of these chromosomes look like the original, we will call each of these non-recombinant. Sometimes we will also call them parental. And I'll slip and go back and forth between them. They mean the same thing. Non-recombinant is a little bit better of a term because it explains what's happening here. Sometimes the parental can be a little confusing because we start thinking about the parents of the individual. That's not what we're talking about. We're just talking about these chromosomes. These gametes, the chromosomes in them, appear the same. Again, remember, they appear the same because we're only looking between genes A and B. Now let's look at what happens here. And let's draw this one over here. It's not involved in the crossing over. We're going to put that like so. That's going to be A and B. And then next, let's write this one on this side. It's also not involved in the crossing over. We call them non-recombinant. Now, the chromosomes here, they're going to look a little different because we have a crossing over event in between them. So it goes big A, comes over here, picks up little b. So this is going to appear as an A, little b. Over here, little a comes over here, and it swaps over and picks up big B. So we have little a and big B. These do not look like the parent, big A, little b. We don't see any chromosome up here that are big A, little b. So we call this recombinant. And same with this one. It's recombinant. If you called these non-parental, that would be okay too. I would prefer that you call these on the end non-recombinant and these in the middle recombinant. It just makes more sense because it's descriptive of what's actually happening. Though we've said this a little of this already, I want to go through parts of this again and make sure we address some of the questions that I mentioned in the note-taking guide. I want to make sure we specifically talk about unlinked genes, I want to make sure we talk about linked genes with no crossing over, and then I want to make sure we talk about linked genes with crossing over. When we talk about each of these, we want to ask, do they assort independently? And we also want to know what the chromosomes look like in their gametes. So let's go over unlinked genes first. Unlinked genes. So remember, these are on separate chromosomes. So let's say we have chromosome 1 here. It's gone through S phase, so we have 2 copies of each. And let's say on chromosome 1 we have gene A. And over here on chromosome 2 we have gene C. They're unlinked, so they're going to assort independently. So let's write that over here. These will assort independent. And our gametes are going to look like this. Remember, each of the chromosomes are going to go into each of the gametes. And if, say, big A goes in this one, and big A could go in this one, and it's just as likely to assort with big C as it is with little c. Likewise, we could have little a here. It's just as likely to assort with big C as it is with little c. We will have a 1 to 1 to 1 to 1 ratio. Each of these are equally probable. Let's erase this. And the next one we want to look at are linked genes without crossing over. So linked genes means they are on the same chromosome. Let's pick a different letter. We have big M and little m. And q and little q. They're linked because they're on the same chromosome. However, they're close enough to each other where we don't get any crossing over between them. We know, and we'll talk about this later when we talk about mapping genes, but I might as well say so now, the closer the genes are to each other, the fewer crossing over events that happen between them. And so in this case, we'll presume that these are so close to each other that there's no crossing over. Because of that, they do not assort independently. They assort with each other. So when you make the 4 gametes, the chromosomes will go into each other. Now remember, there are other chromosomes. We're just drawing this one chromosome because this is the one we're looking at, the one that contains genes M and Q. So there's no crossing over, so M and Q will always be together, little m and little q, and so will big M and big Q, just like that. This, we would give it a 1-to-1 ratio, and we would say all of the gametes are non-recombinant. They all look like the original chromosome. Alright, next I'd like to talk about, okay, now let's look at linked genes with crossing over. So remember, a linked gene, we're going to have our chromosomes like so. They're going to have copied. And we're going to have our genes on here, big R, big S. And on the homolog here, we're going to have little r and little s. And we said, in this case, there's going to be crossing over between these 2 genes. And because of that, when we look at our gametes, let's draw our 4 possibilities here. We're going to have 2 recombinant and 2 non-recombinant. The 2 non-recombinant, big R, big S, they're not involved in the crossing over. And then our 2 recombinant are these 2 middle ones where crossing over has occurred. So you're going to pick up the big R here, crosses over, picks up the little s. So it's going to be big R, little s. And over here it's going to be little r, little s. I'm sorry, big S. What I want to do is take a quick flashback. It's always fun to do. And remind ourselves what unlinked looked like. Remember, unlinked was a situation where we had 2 genes on separate chromosomes. So if these were our genes R and S and they were on separate chromosomes, we might have R on the blue one here, and we might have S on the red one. And because of independent assortment, we would get 4 gametes. You'd get the big R going into the gametes, and you'd get the little r going into the gametes. And then you would also get big S pairing with big R, and you'd get big S pairing with little r. You'd also get little s pairing with little r, and little s pairing with big R. If you notice the way I drew this, up here we have 2 non-recombinant and 2 recombinant. If you just look at the genes in each of these, there's no way to tell these 2 apart. We have big R, big S, little r, little s, just like we have in these 2. We have big R, little s, little r, big S, just like we have in these top 2. How can we tell them apart? Or can we? Well, we can. And we can tell them apart because remember what I told you at the beginning, that this crossing over event in between the 2 genes we're looking at doesn't always happen. In fact, it's rare that it's going to happen between the 2 genes you're looking at. This down here, as I said before, will always be a situation where we have 1 2, 1, 2, 1, 2, 1. If independent assortment is working, we should get 25% of each of these. Up here, because crossing over doesn't always occur, in fact it usually doesn't occur, more of our gametes that are being produced are going to be non-recombinant. So over here we're going to have greater than 25% for these. We don't know how much greater because we don't know how many times the crossing over event occurs. But we know that it's going to be greater than 25%. So they have to be more than 25%. And we know that these, the recombinants, since crossing over is rare, we know it's going to be less than 25%. And a bit of a spoiler alert, when we start looking at distance between genes, that's what we're going to be looking at. How often do we get these recombinant progeny? Alright, well don't tell anybody though. Okay, what I want to do now is write a quick little summary here of what we just talked about with these three situations. I think it's easy to kind of get them muddled in your mind a little bit as you're learning this. With unlinked genes, remember they're on separate chromosomes, we will always get a 1 to 1 to 1 to 1 ratio, where two of them will always be non-recombinant and these will always be recombinant. You could also look at this as 25%, 25%, 25%, and 25%. Okay. Now you can have linked, no crossing over. And in this case, we always get a 1 to 1 ratio. And these always represent non-recombinant. There's no crossing over, you can have no recombinant progeny. And then the last one was linked with crossing over. And remember in that case, so remember on the last slide we said that in each of the non-recombinant classes there would be greater than 25%. So in the non-recombinant classes, if we add them together, we'll have greater than 50% non-recombinant. And likewise, we'll have less than 50% of the recombinant classes. This is what we would predict. A term I should throw out here, when we see this here, a 1 to 1 ratio, where we don't see any recombination, we say that these are an example of complete linkage. They are completely linked. In class, hopefully I ask you this question. Hopefully I remember to. It's an important one. There is another way we can get a 1 to 1 ratio that is not this way. So I'm going to write plus an alternative. And you can think about that. How else could you get a 1 to 1 ratio of only non-recombinant classes like this? Where when you looked at the results, you would say clearly it is completely linked, but upon further analysis, you realize that these two genes are not completely linked. What could be causing this other situation to appear to be completely linked, but they're not completely linked? Think about that, and we'll talk about it in class. Likewise, there's another way we could get data and misinterpret it and say the genes are unlinked, but in reality, they're still linked on the same chromosome. How could we get these two situations? If I don't ask it in class, I'll certainly ask it on an exam, so certainly think about this. So as you're thinking about how to explain these two question marks, draw out the genes on a chromosome and see if you can come up with an explanation. And remember, the only thing Mendel would have predicted from this was the 1 to 1 to 1 to 1 ratio. The others are ideas that he wouldn't have been able to think of because he didn't understand it, or he didn't have the tools that we now have. And remember, the early researchers in this They struggled with it too. So have fun trying to figure these out. And don't get too mad and curse Mendel.
Mendel!
And we'll talk about this a little bit more in class tomorrow. Okay, well that ends this podcast. If you have any questions at all, please let me know, and I'll be happy to help you. I'll see you in class. Bye.